Today, me and my group are going to be presenting um, about meiosis. So to start off with some basics, there is heredity and genetics. So with heredity, that's just the transmission of traits from one generation to the next. Genetics is going to be the scientific study of heredity and inherited variation. Within this, we're also going to talk about gametes, which are reproductive cells. And females are going to have eggs, males are often going to have sperm. They're going to carry their own genetic information, which will be passed along. There are also somatic cells, which are just going to be every other body cell, fat cells, blood cells. These generally won't be passed along or, for reproduction or purposes. Next, we'll talk about asexual versus sexual reproduction. Asexual reproduction is when you just have one parent that is creating identical offsprings or clones. This is usually a very quick, low energy process that will create lots of offspring. But with that comes the fact that there will be low genetic diversity, and if that parent has a disease, those offsprings are more susceptible to those same diseases. During sexual reproduction, this often happens between us humans, a male and a female, coming together, creating a child. You're going to have two parents, and that offspring that they have is going to have a unique genetic combination. And this is good for genetic diversity and for better chances of survival. Because there's two parents, that child may not be as susceptible to certain diseases like one of the other parents. The only downfall to this is that it does take more energy and there are fewer offspring that can be made as opposed to like asexual reproduction where they just can make. So now that we've got that out of the way, what is meiosis? Meiosis is a type of cell division that reduces chromosomes number by half, creating haploid gametes. So you're going to have your initial cell, which is going to be that diploid cell, and it's going to have all 46 chromosomes. When it goes through the process of meiosis, you are going to be left with two separate gamete cells, and they're going to be haploid cells, and each of them is going to have 23 chromosomes. So there are two phases of meiosis. We're going to start with phase 1. So both of them are going to have prophase, metaphase, anaphase, telophase, but it's just going to be phase 1 and phase 2. So phase 1 of prophase, you're going to have those chromosomes becoming visible, pairing up, sharing that genetic information, and then those chromosomes are going to align at the metaphase plate or the equator of the cell, and they'll begin to separate to opposite poles. Then you'll go through your phase of phase 1 in cytokinesis, and from that diploid cell, you're going to have your two haploid daughter cells form. Then those two daughter cells will begin to go through the second stage of meiosis. And so in those two daughter cells, you're going to have the chromosomes condensing and becoming visible and moving toward the equator. Then through metaphase 2, they'll begin to align at that equator. And then in anaphase 2, those sister chromatids are going to be separated, as you can see to the right. Then the last stage, telophase 2 and cytokinesis, you're going to end up with 4 genetically distinct haploid cells that are formed. So you get your diploid, then you're going to get your 2 haploids. Then from those 2 haploids, you're going to get a set of 2 haploids from each. It's just a whole chain of breaking down. So from here, I will let Abigail take the rest.
I'm going over the remainder of the slides. So first starting with crossing over and genetic diversity. Homologous chromosomes exchange genetic material in prophase 1, and then on the right here you can see where it says prophase 1. This creates recombinant chromosomes combining maternal and also the paternal genes. And a good analogy that we used for this is it is like splitting a book into two, rewriting parts of both of them, and ending up with two unique stories. The next slide is genetic variation. So there's three kind of topics that we wanted to cover. So independent assortment, the homologous chromosomes align randomly in metaphase 1. And then the second would be crossing over. This is where the genetic material is exchanged between homologs, creating new combinations. And then number 3 would be random fertilization. This is just any sperm can fertilize any egg, which adds to the variation. The real world example that we used was genetic variation in humans. So genetic variation helps species adapt to challenges like diseases. An example of this would be during the Black Death, also known as the plague. Genetic variation allowed for some humans to survive as certain individuals had mutations that made them resistant. So next are the key questions. So what is heredity? Heredity is the transmission of traits from one generation to the next. And what are gametes? They're reproductive cells that carry genetic information. The sperm and eggs is kind of like an example of that. What is the difference between asexual and sexual reproduction? So for asexual reproduction, it's just one parent and identical offspring or clones. And then for sexual reproduction, you have two parents, offspring with unique genetic combinations. And there are pros and cons to both of those different reproductions. How does meiosis contribute to genetic variation, independent assortment, crossing over, and also random fertilization? What are the stages of meiosis and what key events occur? So since there's technically like, I guess you could say, two stages of meiosis. In meiosis 1, you have prophase 1, metaphase 1, anaphase 1, and telophase 1, and cytokinesis. And then in stage 2, you have prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Thank you.